One, and you're on. Hello, everybody. Welcome to the Biotechnology and Society webinar series, co-sponsored by Harvard Medical School Center for Bioethics and the Museum of Science here in Boston. I'm your host, Dr. Insoo Hyun. I'm the director of the Center for Life Sciences and Public Learning here at the museum. This series is meant to bring together the cross-section of biotechnology and research on the one hand, and the public's interest in these areas on the other hand. And this is really a forum for public science learning where people get a chance to not only hear from leading experts in the field, but also to ask them questions online and to get those questions answered in real time. Today, our topic is stem cells in space. And my guest is Dr. Arun Sharma. He is an assistant professor at the Cedars-Sinai Medical Center in Los Angeles. And that's where he uses human stem cells to study cardiovascular biology and disease. I'm just so excited about this topic because it's really out there. It's really out of this world, so to speak. Arun is just a fantastic guest. I have two things I just want to highlight about him. One is that he was listed under the Forbes 30 under 30 in science list. It's very cool. And he's also a co-host of the Stem Cell Podcast, which is an internationally acclaimed platform for science learning around the world. Now, you will get a chance to ask Dr. Arun Sharma your questions along the way. And to do that, please go to slido.com. Our site is Slido, MOS webinar, and under that title, you can submit your questions, and I'll be moderating those halfway through the hour. But without any further ado, I'd like to bring on Dr. Arun Sharma on board. Welcome. Great to have you.
Thanks, Insoo. Great to see you.
Yeah, so I think before we jump into the exciting work you've been doing out in outer space, and I just wanted to just maybe hear a little bit more, let the audience hear a little bit more about the kind of work you do at Cedars-Sinai, maybe some of the work you did before your space work. I'm really fascinated by this idea of using human stem cells and then de-differentiating them or driving them down the pathway to study, let's say, heart cells and cardiovascular function. So maybe tell us a little bit more about your scientific background and your work in stem cells more generally.
Sure, I'd be happy to talk a little bit about what we do here at Cedars. I'm a stem cell biologist and also a cardiovascular biologist by training. I got really interested in studying the heart in large part because the heart is one of the organs in the body that has a very limited regenerative capacity. It's not able to regenerate itself very well after an event such as a heart attack, for example. And this is an interesting thing to study in large part because there are so many exciting new developments in stem cell biology, such as induced pluripotent stem cells, which allow you to mass produce human iPSC-derived heart cells from a small sample of someone's own skin or blood. That's what really got me excited in the field in the first place. That's why I went to grad school to do my PhD in stem cell biology. And now in my own laboratory, I use these stem cell-derived heart cells to study cardiovascular disease in dish, understand why certain cancer drugs can do damage to the heart, understand how certain diseases of the heart arise during development, such as congenital heart disease that unfortunately many people are born with. So these are just very powerful cells to work with, and this is really the focal point of my lab.
So, Arun, how do people study your field before stem cells arrive on the scene? Like, what typically do people do, and how does stem cells sort of change the game of your research or research in this area?
Yeah, so the power of these stem cells is that for the first time, really, you can mass produce human heart cells in a dish by the billions using differentiation from induced pluripotent stem cells. And before this, there have been animal models which have been around for a long time, such as mouse models and various other animal models. But there wasn't really a great way to mass produce human heart cells until we were able to do this from pluripotent stem cells or induced pluripotent stem cells. And now that we can mass produce these cells, we can use them for a variety of other purposes, such as for drug screening, identifying new drugs that may be able to help the heart and, study various forms of cardiovascular disease in that way. And also for other exciting projects, such as looking at the effect of microgravity on the human heart. Because we can make these cells by the billions, there are so many different things that we can do with these cells.
Yeah, well, this technology is still relatively new in the biology world. What drives someone to want to then explore this stem cell biology and all the things it could do off of Earth? I mean, isn't there enough to learn here on Earth and labs, on the ground? Why would you want to go out into outer space with some of this stuff?
Yeah, absolutely. Well, the reality is there's still so much we don't know about how the human body actually works in space. And certainly we've been in space for a half century plus now. But we've gotten an understanding of what actually happens to the tissues and the organs of the body. The heart, for example, changes shape and size over an extended period of time in low gravity. And this has been demonstrated in astronauts for decades now. But we still have less of an understanding of what actually happens to the individual cells of the human body. How did the blood cells respond to being in microgravity in space flight for an extended period of time? What about the cells of the brain? And we do know that the muscle cells and the muscles and the bones of the body degrade pretty quickly after astronauts are in space for an extended period of time. And in fact, one of the main things that astronauts do every single day to stay healthy is to exercise their muscles and their bones just to do that resistance exercise to stay strong. But cells comprise everything. Cells comprise the muscles, the heart, the bones of the body. And so we want to get a better understanding of what's actually happening at the cell level to the human body in space. And that's why these stem cells and stem cell derived heart cells, which is what I study, are really a powerful tool to help us understand what's happening to the body in low gravity at the cell level.
Mm-hmm. So before these cells got taken up into outer space, what were some of the hypotheses? Like, what did you think you might learn, or what were some of the unresolved questions you wanted to explore by taking them out there?
Yeah, I think initially we wanted to see if those tissue and organ level observations that we've seen over the years, for example, the heart changing shape and size and not beating as hard, if those same observations would be extended to the cell level as well. Because, there are billions of individual heart muscle cells that comprise the heart. And our hypothesis, though, is that we might be able to see some of those changes even at the cellular level. And indeed, that's exactly what we saw. And it was very exciting to see, because even at the cell level in our experiment, which we conducted in 2016, we saw that these cells were actually not beating as hard in microgravity. And if you want to pull up, I guess, the first video, we can actually show you what some of these human heart cells actually look like in a dish when they're contracting. Yeah, so this is what our human iPSC-derived cardiomyocytes or stem cell-derived heart cells normally look like. They're beating in a dish pretty regularly. And we saw that in our spaceflight experiment, these cells were actually not contracting as hard. And that was really exciting, because that's parallel to what we're seeing at the organ level.
on. Beating cells. Sorry about that, folks. I think there was a technical challenge here at the museum, but I think we're back on now. So Arun, when we dropped off, you were talking about taking these cells up to space, and we were showing a video. So can you just walk us through that video? And let's get back to the video on the screen, please.
Sure. This is a video of beating stem cell derived heart cells as they would appear in a dish on the ground. And you can see that they're contracting, beating pretty rhythmically. And this is what we see every day in my lab. We mass produce these cells by the billions. It's really fun to work with these things because not a lot of cells move in real time, which is really exciting to work with. And so these are the cells that we actually sent to the space station to actually examine how microgravity was impacting their function. And like I mentioned, we were seeing changes that were happening at the cell level that paralleled with what happened at the organ level, mainly that these things were not contracting as hard. A compensation, a compensatory response, as we call it, to the effect of microgravity.
I thought of all the stem cell work I've seen, whenever I see heart cells beating derived from stem cells, that's really the most exciting because actually they're doing something in the dish and they look pretty incredible. So tell me a little bit more about what you're seeing in the space experiment. What do you think is going on there?
Yeah, we think that these cells are sensing the low gravity environment in a way, and they're adjusting their function to that low gravity environment, not only at the genetic level, the gene expression changes, which is something that we actually looked at in this experiment, but also functionally. As we're seeing, these are really active cells. They require a very strong metabolic activity to actually contract. The heart is one of the most metabolically active organs in the body. And so these changes are happening not only at the genetic level, but also at the metabolic level, the functional level. And this is something that happens within a matter of days, really. So it tells us how plastic and how adaptable the individual cells of the human body could be to the stress of microgravity.
Yeah, so I want to return a little bit more to some of the really great things you're learning with these experiments, but let me just zoom back a little bit further. Tell me more about how you got involved in this work. I mean, you're doing work like many other people in your field, just in labs all over the country. And what inspired you to take this next step? And the other question is, are there other people doing this kind of work, looking at stem cell biology in outer space and microgravity? I'm really kind of curious about the shift from kind of more conventional stem cell biology and research in labs on the ground, and then just taking it to this whole other level.
Yeah, so I guess to answer your question of how I got involved in this in the first place, I grew up as a big space fan, as I think a lot of us do. I actually grew up in a city in Alabama called Huntsville, which is our one claim to fame is that there's a space center there. There is a NASA base there. So I was really excited about space from a very young age. I also got really interested in biology as well, and I ultimately figured out a way to intersect these two passions through this project that I first did in grad school. That's how I got into the field, and really, over the last decade, space research has become so much more accessible to not just the stem cell community, but the greater biomedical research community. And this is in large part because because of the advent of commercial space. There are companies such as SpaceX and all of these other private enterprises that are able to launch rockets more frequently these days. And that means we can send experiments more and more frequently to the International Space Station. And the next step is actually to have completely independent space stations that are off of the International Space Station that could potentially only be devoted for biomedical research purposes. And again, this comes back to the question of why we're doing this in the first place. There's so much we don't know about how the human body actually works in space. But the other part of this is, could we actually harness microgravity? Could we use microgravity to grow cells and biological products in a way that's simply impossible on Earth? And there are examples of this in material science. For example, there are certain fiber optic cables that can only be manufactured in microgravity because the microgravity presence gives the cables a quality that's just simply orders of magnitude higher than what you can get on the Earth. And what I'm really excited about is in the next 10 years, we might find that quote killer app for biomedical research as well. Perhaps we can make a cell type or manufacture cells in a way that's just simply impossible on Earth. And so that's what really excites me about this work.
So how did some of these initial projects get started? I mean, like who funded it? How did you pull the proposal together? I mean, it's really, quite remarkable. It must have taken quite a large interdisciplinary team. Tell me more about sort of the beginnings of this project and how you actually got up there.
Yeah, absolutely. It's definitely an interdisciplinary team that's involved in this project. It's not just cell biologists, but mechanical engineers, aerospace engineers, folks from NASA who ultimately, they're the ones who funded the work. It's not easy. This is a complex experiment. In large part because you can't grow cells in the same way in microgravity that you can on the ground. Here on the Earth, you grow your cells in typically cell culture dishes, and you can take off the lid of these dishes, and you can change the media, you can manipulate the cells however you want. But if you do the same thing in microgravity, you might run into a problem. If you open that culture plate, the media might escape into the space station, and that's definitely not something the astronauts want. It's not something NASA wants either. And so actually to get around this problem, we worked with a really talented group of people, a team of mechanical engineers at the University of Colorado, Bioserve Space Technologies. And what they did is they actually designed some custom pieces of hardware for us to actually grow our cells in. And if you want to pull up the next set of images, I can show you what that looks like. Well, actually, before we get to that, this is actually a picture of me at the launch, which is a very exciting thing to be a part of. I guess we can move on to the next one as well. This is our group. This is our group actually preparing the cells that we were going to introduce into these custom 6-well culture plates. This was me and Dr. Peter Lee, who is a co-investigator on this team. This is the groundwork that we were doing right before the launch, right the day before the launch at Kennedy Space Center in July 2016. It was a nerve-wracking experience because it was 3 years of work, cell culture work, that was all leading to this moment. This is me just very nervously looking at my cells with the microscope,
pretty nervous there.
sure yeah, not a lot of smiles right there. Very focused, making sure that the cells are looking good.
You also look like you're like 12 years old. Like, when was this?
Yeah, this was not 8 years ago or so.
Okay.
So I got a little bit more facial hair now, which is I guess a good thing. But I think in the next set of images, you'd be able to see, yes, that right there. So this is actually how the cells were grown. This is that custom piece of hardware that I was talking about that the team of mechanical engineers put together for us. This looks familiar to a lot of folks who are doing biomedical research. We grow cells in 6-well plates, as we call them. But the cool thing about these plates, you can see every single separation, every single well is individually separated from the other. And this means that we can grow our cells in a way that's fully enclosed. That means the cells themselves won't escape into the space station, which is really critical. And in the worst case scenario that something happens to one of the wells, say C1, because each of these wells is isolated from the other, that contamination or whatever bad thing that might happen to one of the wells, it won't spread to the other well. So this is a useful way of growing these cells aboard the International Space Station. And this is, I would say, for the most part, how people actually do research, biomedical research in space in these custom pieces of equipment.
Mm-hmm. What other customization do you think is necessary to move some of this forward?
Yeah, there's a lot of work that we have to do to really improve the throughput of these experiments. So being able to do more and more of them again and again. It's easier to get into space and do this kind of research these days because, like I mentioned, it's cheaper to fly to space and more commercial partners are getting research up there. But it's still tricky to actually conduct the experiments. So there's certain advances in robotics that we can take advantage of so that we may not even need to utilize astronaut time to do some of these cell culture experiments. Astronauts are really busy. They're maintaining the space station. They're doing a lot of experiments. But if we can, automate some of these experiments, that would be very useful. And if you pull up the next image, I think this is an image of Kate Rubens, Dr. Kate Rubens. who is the NASA astronaut in charge of actually maintaining our cells in orbit in these custom pieces of hardware that I was talking about. Now, it was such a pleasure working with Dr. Rubens, because not only is she just an amazing astronaut and NASA astronaut, she's also an accomplished cell biologist. She was actually a principal investigator, a lab head at the Whitehead Institute in Boston,
Wow.
just down the road from the Museum
Uh-huh.
of Science, before she just decided to apply for the astronaut program and become an
What an
astronaut.
underachiever. Wow.
I know, right? So we were really lucky to be working with her, because as a current and fellow biologist, we could speak the same language. So if
Exactly.
she was able to make an observation in our culture plate, she could tell us, oh, the cardiomyocytes are beating a little bit less, and she could tell us how these cells are actually changing over time. And we were speaking that same language, so that was really cool. But she's also, like I said, really busy, not only doing this experiment, but also doing so many other experiments at the same time. So figuring out ways to automate some of these approaches and these experiments would be really useful.
So the papers that come out of these experiments, do the astronauts get on the author line as well?
Yeah, they do actually.
That's
So
pretty cool.
Kate was, in fact, one of our co-authors, and that was pretty cool.
That's great. So tell me a little bit more about how long they have to sort of be up there for you to get your research results. As you know, research on the lab on the ground could take weeks, months, could take a really long time. How long are you able to study the cells up in space?
Yeah, it's a great question. This is something that is variable, and it depends on the experiment that you're doing. For example, the research that we were doing in 2016, it was a 1 month long experiment. And that in part has to do with when the astronauts are able to go up, when things can go up and back down in terms of experimental hardware. That's why our experiment was limited to 1 month. There are other experiments, and in fact, we have a launch coming up and another project coming up here in May that's going to be a 10-day mission. And that's in part, again, because of mission constraints. The mission itself is a 10-day mission, so we have to adapt our science to those 10 days. But the other exciting advance, if you're looking to the future, like I mentioned, a lot of these experiments are getting automated. So you can envision that perhaps you can send an experiment up to the space station and then just let it go and let robots or automated machinery handle and take care of those cells for even months at a time. And that would give us, I think, really exciting new pieces of data to think about.
As a bioethicist, I can't resist asking you a bioethics-like question, but don't worry, it's an easy one. So the cells you send up there, those are derived from induced pluripotent stem cells, right? So these are stem cells that are created from skin cells or other, somatic cells from a patient or a person. Do the persons who provide the iPS cells, the original donors, do they know that some of their cell lines have gone up in space? Do you have any idea if people are aware of that as a donor?
Typically, no. Typically, no, because when we make those initial informed consents, we can say that broadly these cells will be used for research purposes. We don't always specify what those research purposes are, whether it's growing the cells on the ground or in space. But there have been situations where we know exactly. We going into this project, we had informed the person that these cells are going into space, and that they, in an essence, in a way, they were going to be an astronaut, which is kind of exciting to think about. Even if you yourself aren't going up in there doing research, your cells are contributing to science in space. So it does go both ways.
Yeah, I was just wondering that because I think many people would think it's pretty cool if their cell line got up there and participated in the work that you're doing. So you seem to suggest, or you did suggest, that there's a way to maybe use microgravity as a to your advantage for manufacturing. And tell me a little bit more about that. So what are some of the aspirations for, in the cell biology world or biotechnology world of one day maybe using space as a site for getting things that you cannot create on the ground?
Yeah, I think this is one of the most exciting things about this area. It's a huge unknown about how we can harness microgravity for cell manufacturing. We know for a fact that stem cells behave differently in space. They proliferate differently, they differentiate differently, and we have additional experiments actually lined up this year to further flesh that out. But one exciting possibility is manufacturing tissues in a way that's simply impossible on Earth. And I'll give a case example of this. 3D printing is something that's really taken off, pun intended, I suppose. There's actually been a 3D printer that's been on the space station not too long ago. And what they're able to show is, by printing certain biological materials, you can do that in a way in microgravity that's just not possible on the ground. Sedimentation properties are totally different. Fluid dynamics are very different in space. And so you can harness some of these properties to 3D print materials in a really unique way. Going outside of just, bioprinting, that sort of thing, I would say currently the first and still most perhaps most rewarding killer app that's been developed so far in space has been in the area of protein crystallization. Now, this is an area that's really important for drug development and drug discovery. Pretty much any drug that's out there on the market now has to be appropriately crystallized so that you can see its structure and its form, so you can see how it's interacting with other parts of the body and that sort of thing, and other drugs. And there have been situations where we can only create the protein crystals of certain drugs in space. And the quality of those protein crystals have been so far above anything that's been possible on the ground. So that's, in my mind, so far the best killer app for microgravity when it comes to biomedical research. But for cell therapy and for cell manufacturing, tissue printing, I think the next 10 years are really going to tell us a lot.
Are there industry partners that are interested in this work at this stage? It's still pretty early on, but tell me more about that.
Absolutely. And you can envision a situation where, say, there's a protein, a drug that's only able to be crystallized in space. That's very exciting from an industry and commercial perspective. Big pharmaceutical industries would be rapidly involved in this sort of area if we're able to find that situation. And certainly there have been talks with commercial entities, with the commercial and private space stations that are coming online soon. Certain pharmaceutical companies are wanting to get aboard those space stations to carve out a research enterprise on those private space stations, because the other part of this is intellectual property. If there's a drug that's able to be discovered in space or formulated in space that's far superior to what you can do on the ground, then from an intellectual property standpoint, I think a company would want to wrap some IP very quickly around that area. And so this is, like you mentioned, this is just starting to emerge, but indeed, pharmaceutical companies are starting to get involved in this space. Even smaller biotech startups are getting involved in this space as well.
Are there other countries interested in getting into the space? And there's sort of like a budding space race to do this?
Definitely, you could call it a space race in the biomedical research realm. Definitely, there are other countries around the world that are, sending private space stations, utilizing their own space station. China, for example, has their own space station that's going online and has been online for a little while. And so, really, I think it's a bit of a race to see who can figure out those biomedical applications in microgravity first, and figure out those quote killer apps that I keep on referring to.
Yeah.
I think the next 10 years are going to be really telling in this area.
So where does the U.S. federal government stand on this? Are they enthusiastic? Are they aware of the possibilities? Tell me a little bit more about like the government's response.
Yeah, I think well, for one, the U.S. federal government has traditionally been the greatest funder of space-based research. I do think, as I mentioned, commercial enterprises are coming online, but in large. A big reason why the U.S. federal government is funding research in space is because, the government wants to maintain a strong leadership in the international community when it comes to space-based research. We want to make sure that the United States is maintaining our leadership in space, and as we have for many decades now. And as a case point in that, I was actually recently invited to a National Space Council meeting in Houston, Texas, chaired by Vice President Kamala Harris. And I think if you want to pull up one of the images, we can show an example of what was going on at the meeting. This is me sitting in front of the vice president and also the head of NASA on the right and various cabinet members. It was almost like a Senate hearing. You could see me sitting at this little desk being
you weren't in trouble,
interrogated
though.
by all these government officials. It was definitely nerve-wracking.
Yeah.
But the reason I was there was I was giving an address as to why we should maintain our leadership in space-based research and what's the actual importance of doing biomedical research in space. And so I gave some examples that, some of those I mentioned here on the show of like protein crystallization, 3D printing, all these applications that are just starting to emerge and that we're just starting to figure out more about. But it's a point of emphasis for the federal government, definitely, to keep funding this work so that the United States can maintain our leadership in this area.
That's terrific. We're getting lots of questions coming in on Slido. So just as a reminder to the audience, you can go to slido.com. And we are MOS webinar under that heading, hashtag MOS webinar. And please submit your questions live online. So let me read a few of these and let's start getting into the questions here. This is from Anonymous. Does the study of stem cells in space have any impact on future healthcare in space travel? Will this enable us to treat people in space? Interesting question.
That's a great question. And indeed, this is a really exciting area of study. Like I mentioned, if we can show that we can manufacture stem cells and their products in space in a far superior way than we can on the ground, then I think that has a lot of implications for astronaut health. Say in the situation that, we're going to Mars, and this is something that we will do over the next few decades, right? Mars and the journey to Mars is not an overnight thing. It's something that takes months to do. And once you get to Mars, that's a presence that could take a few months as well. And so long duration missions require potentially a next level of medical therapy and medical treatment. In the situation, perhaps if an astronaut gets injured on the way to Mars or in Mars, perhaps we're able to regenerate some of those, some of their tissues, some of their tissues using stem cell derived products that were manufactured in space. And so this is an area that not only the US is investigating, but other countries around the world are investigating, and private partners, commercial entities are investigating as well. So it's a very valid question.
Yeah, you mentioned, the long journey to Mars. Do you know if there's any interest in studying human germ cells, sex cells in space, and kind of like study very early embryo development? I assume if somebody got pregnant in space, would that have any negative effect on the developing embryo?
Yeah, this is a really interesting area, and I'm sure as a bioethicist, you are very interested in this area indeed. Of course, if we're spending an extended period of time in space, human reproduction is something that we have to discuss. And animal experiments have been conducted in this area for many years, but this is definitely an area that folks in the stem cell community are very excited about. Over the last couple of years, as I'm sure you're very well aware of, Insoo, there have been a number of studies in early embryo development that have been emerging, and a lot of exciting technologies in stem cell biology that enable us to study human embryo development at a high resolution time for the first time, really. And I think intersecting some of these new technologies, such as blastoids and early embryo models with space science, I think is inevitable. I've actually been hearing rumblings of folks who are in the stem cell community who are hoping to do that exact experiment and perhaps even send some early human embryo models to microgravity. But certainly it's an ethically charged area, a lot of regulatory hurdles to get around perhaps, but it is an area of interest.
Yeah, just to add a little more commentary to this issue. So what we're talking about here, folks, are stem cell-based models of very early human development. So you can get, iPS cells from a living person or from, a tissue sample, and drive this down to create this sort of like three-dimensional model of very early development after fertilization. Now, these are not complete human embryos because they don't have all the aspects of a real human embryo, but they have some parts of it. So you can really kind of focus on particular aspects like yolk sac formation or particular germ layers. And so it'd be really fascinating to take some of these models up to space to see what happens in microgravity, because you would want to know if there's any harmful effects of space travel for human development, again, in the event that somebody conceives in outer space. And so it could be really fascinating to see what differences you might see in how the cells behave, how they self-organize, how they continue to develop in a healthy way or not, before you actually go up there with real human embryos during some kind of long travel. So I think it could be really valuable information. Now, another question came in, Arun. Somebody is asking, do you have any ethical issues or ethical concerns in your own work? So I'm kind of curious to know if you have any in mind. If you don't, that's fine. But if you have any, love to hear it.
Yeah, actually, even before we get into that, I think the previous question is very interesting. There's actually one other element that I wanted to touch on there. This is the element of radiation in space, because if we go towards missions to Mars and beyond, this becomes a much more serious issue. When it comes to low Earth orbit and work that's being done on the International Space Station, the impact of space-based radiation is not as significant as it would be if we took an extended mission to Mars. And that's in part because of the protective environment that the Earth's magnetosphere and the Earth provides to the low Earth orbit. But if you have a situation in where perhaps somebody in space is trying to conceive, then you can envision a scenario where enhanced radiation exposure could alter development in a way. And so this is something that's, I think, definitely worth further study down the road using some of these early embryo models that you're alluding to. When it comes to the ethical considerations of my own work, I think perhaps not as many as in comparison to some of the early human embryo modeling work, which I think is inherently charged with some of those ethical considerations. I think one thing to make sure is that the studies that we're doing are representative of the greater human population and the diversity that we actually see in this country, for example. So ensuring that the stem cell derived heart cells or any iPS or stem cell derived cells that we're examining, we want to make sure that those are representative of the greater population. And so what that means is taking stem cells and iPSCs of people from a variety of different genetic backgrounds to outer space to really see the impact of microgravity on stem cells from these different individuals, from different genetic backgrounds. I think that's something critical that we have to integrate into our work.
Mm-hmm. I wonder if you ever have concerns along the lines of the following. We know that there have been some high-profile cases of people who reported inaccurate research results, to kind of put it mildly, or fabricated some of their research results. So a lot of people now think it's not until somebody reproduces the work that we could really kind of call it like scientific canon at this point. Your work would be extremely hard to reproduce. How do you assure journal editors, how do you assure peer reviewers, the greater scientific public, that they could trust your research results, given that it's so hard, if not impossible, for other teams to replicate your work?
Yeah, this is a really good question, and is a major struggle in this area. I think initially, yes. I mean, this is because this work is so hard to reproduce. We have to take some of the results that we're getting with potentially a grain of salt, right? And like you're alluding to, the best scientific work is the work that can be reproduced by anybody, any lab around the world, and not any lab around the world has access to a microgravity environment. That's just the reality. One possibility is being able to reproduce some of these experiments on the ground through microgravity simulators. There are ways that you can simulate microgravity on the ground through devices called clinostats, magnetic levitation, different approaches. But this is not a perfect replicate of the true microgravity environment that you would see in low Earth orbit. Mm-hmm. So I think going back to one thing I had alluded to at the beginning of the talk, automation is a very important way and a very important approach that we can take to address some of these issues of reproducibility in science. not just science on the ground, but definitely science in space. Because if we can take out, quote, the human element in some of these experimental approaches, then perhaps these studies can be more and more reproducible. And again, like I alluded to earlier, astronauts are really busy. They have a lot of things going on. This would potentially make their lives a lot easier as well in terms of automating some of these experimental approaches, increasing your N values, as we like to call it in the biomedical research community. This is, I think, the inevitable next step for this biomedical research in space.
Yeah, we have just over 15 minutes left. So audience members, if you have a question, please go to slido.com and ask them if there's any like experiment you think would be really cool to run in space and you want to know if it's that even possible. Is it on the radar of people like Dr. Arun Sharma? Please ask that question. I would love to know, hear from you, what kind of knowledge you hope that this kind of work could unpack. So let me ask you a very naive question, Arun. So is there a difference between microgravity and zero gravity?
There is, and zero gravity is it's something that's a little bit more that's not as likely, right? Because no matter where you are in the universe, something is going to be exerting a gravitational force on you. So zero gravity is true zero gravity. I don't think is truly possible. Microgravity is something that we experience in low Earth orbit. So the International Space Station is only a few hundred kilometers actually above the Earth's surface, and people may not realize that it's potentially even closer for me to go to the International Space Station than it would be for me to go from Los Angeles to the Bay Area, for example. Yeah. So microgravity and zero gravity, there is definitely a very important distinction there.
So that gets us into this next question that came in online. From a cell biology perspective, is there any expected difference between the effects seen in low Earth orbit versus deeper space?
Yeah, that's a fantastic question. I think referring back to something that we had discussed earlier, a major issue when it comes to deep space is radiation. Radiation is something that, as we know, has a major impact on biological functions, on DNA replication, on cell replication. And this is definitely an area of needed study. And in fact, there was the Artemis 1 mission, which was the first, which is part of the Artemis program, aiming to return back to the moon for the United States. The first rocket, the Artemis 1 rocket that was actually going around the moon just a few months ago as the initial launch in that mission, it actually carried a sample of yeast cells on that rocket. And this was done in part to see what's the effect of deep space radiation on yeast cell replication and that sort of thing. So this is an area that's, I think, going to get a lot more interest and is going to be worth fleshing out in the years to come.
I have a few questions that have come in that are a little bit more heart cell specific for you. How do you actually quantify the differences in the behavior of heart cells in space versus on Earth?
Yeah, that's a great question. And this has to do with experimental controls, which is something that, no matter what sort of biomedical research you're doing, you have to deal with experimental controls. So being able to compare your experimental condition to an unchanged condition. And in our situation, whenever we're doing a research project in space, we always like to have a set of ground side controls, as we like to call it, a parallel experiment that's happening on the ground. And that would help us examine what are those actual true differences that are caused by microgravity. It's definitely not perfect because there are a lot of other variables other than microgravity that could be influencing our experiment in space, but having that parallel set of ground side controls is very important.
So currently you've observed the heart cells with regard to how they change in microgravity. What else do you quote unquote expose the cells to, if anything?
Yeah, so other things we can expose the cells to include certain chemicals, certain molecules that may be able to enhance their function in space. So in our initial project, we're able to show that the cardiomyocytes, the heart muscle cells, don't contract as hard in a microgravity environment. And this has, implications for astronaut health, but the next step is being able to reverse some of these phenotypes, being able to restore proper heart function, perhaps in space. perhaps we can develop in combination with, in collaboration with the pharmaceutical industry, a drug that can improve cardiac function, not only at the cell level, but at the organ level, and that perhaps astronauts could take during those extended missions to Mars and beyond to keep their hearts healthy. Perhaps there's even something comparable that we can develop for the bones to prevent bone loss or prevent muscle loss. So I think that is the next step, and NASA is very excited about this area, developing what's called countermeasures to space-based microgravity and radiation exposures and so on.
So it seems like the cells you take up to space are all quote unquote healthy cells, or would there be any reason to take disease-specific cells up there in the future?
Yeah, I think it's a valid point, because I think as we go towards the future, folks from all different backgrounds are going to be going into space. These days, astronauts are typically quite healthy, very healthy young individuals who are going into space. But you can envision when spaceflight becomes cheaper and cheaper, folks from with various disabilities or with other muscle degradation phenotypes might be going to space as well. And so that could be interesting from a quote disease modeling perspective, taking stem cell derived heart cells from somebody who has, say, a cardiomyopathy or a heart muscle defect. We could study how those iPS derived cardiomyocytes from the cardiomyopathy patient may function in microgravity. Perhaps that muscle degradation phenotype that we're talking about, it happens faster in those disease specific cells. Because, and this is again in the area of disease modeling, being able to predict if an actual person with that disease phenotype actually went into space, perhaps we can make that parallel from that person's own cells as well.
So this next question must have been inspired by your comment on yeast, but somebody asked, can bread rise in space? That's kind of a fun question.
Can bread rise in space? I actually don't know. Maybe it'll rise in a different way, kind of in a spherical way or something like that. That's a great question. I don't know the answer to that.
Yeah, bread in space. Actually, speaking of, I mean, is there any sort of interest in understanding food production in space? Because you're talking more about, using space as kind of, to understand better ways of maybe developing drugs, et cetera. But I would assume at some point, if we ever really get serious about space travel, I mean, I don't think we would always want to have packaged food from here on out for anybody going. I mean, what about people who are interested in, like, I don't know, is there plant work being done in space, or it's something kind of, again, non-human biology related?
Mm-hmm. Yeah, this is something that NASA is looking at very closely. And in fact, I'm a judge for a NASA challenge called the Deep Space Food Challenge.
Oh,
and
wow.
This is exactly what folks from around the country and around the world are hoping to figure out, figuring out the most cost-effective, efficient way to grow plants, various other types of protein sources for extended periods of time in microgravity. Because this is exactly what we need to address in those deep space missions to Mars and beyond. We can't feasibly keep sending shipments of pizza from Earth, every month or so. People have to be able to grow their own crops, their own plants, healthy, nutritious food in space. This is definitely a big area of study right now.
Yeah, I mean, I think while we're somewhat free associating here with some of these questions, I think a question I have for you is, there is some interest amongst conservation biologists. It kind of seems out there, but I think it's feasible to put up a biorepository of endangered species cells and DNA up on the moon or someplace in orbit away from Earth in case something catastrophic happened, nuclear disaster, whatever. Do you think, I assume these are going to be frozen down, but do you think having those kinds of animal cells, non-human cells up in space in hopes of future biopreservation, are there challenges that microgravity have on those kinds of cells? I mean, they're not dividing right now, that they're just in storage, but I'm kind of wondering, in light of what we've been talking about, is there work that needs to be done to make sure that that's actually a safe place to store specimens?
Yeah, it's a fascinating question. I think, this is it's an interesting area of study, and we certainly have biorepositories on Earth. The seed bank, for example, that's, I believe, found in the Arctic area for preserving seeds from different species, different plant species in the case of a nuclear event or some other catastrophic disaster. And so envisioning a scenario where we could have some sort of gene bank in space, could that be possible? I think it's something worth looking into, but I again think the issue of radiation is a big one. Even at the moon, the exposure to radiation is orders of magnitude above what you would experience here on the ground or in low Earth orbit. And so could that detrimentally impact cell division, even if these are frozen samples? Could that impact the DNA of these frozen samples? It feasibly could. So that is a major consideration to think about.
So I love this next question. Arun, as a research scientist, are you ever frustrated that you can't go up there with your experiments?
Oh, yes. And let me tell you, I have tried. I have applied to the astronaut programs, maybe trying to follow in Dr. Kate Rubin's footsteps, but I have not been successful as of yet. I'm going to keep trying. But yeah, I do think there's a need for biomedical researchers who are also astronauts, because as we're talking about in this discussion, biomedical research is becoming more and more accessible in space. More and more people are doing it. And I think there's a need for astronauts who can speak that same language, who know how to do cell culture on the ground, and perhaps are able to, do that, be the point person for doing biomedical research in space. And I think private enterprises and commercial partners are definitely looking into this. These private space companies, for example, there's there's a company called Axiom Space, and their mission is to create the first private space station, and they have a mission being launched in a couple of months in May, their Axiom 2 mission. And one of the astronauts on that mission, a private astronaut, is actually a biomedical researcher from Saudi Arabia. She doesn't have a traditional astronaut background. She doesn't have a military or an aerospace background, but she is a trained biomedical researcher and who's been doing biomedical research for a decade plus. And so that does tell you that, yes, I think in the future, there could be a need for somebody like me and actually doing my own experiments in space. But for right now, I'm here on the ground, and I'm going to do my best that I can from down here.
Is there on the ground training that you could be involved in? So I'm just wondering, like, how similar can you simulate what would be going on in space here on the ground? How would you do that? And would there even be a need for that kind of prep work?
Yeah, it's something we alluded to earlier in the conversation. There are ways to simulate microgravity on the ground through random positioning machines, magnetic levitation, this quote unquote vomit comet that people have heard about, where you can go on a parabolic arc in an airplane and simulate a microgravity environment for a few minutes. I know folks who have been on those things, but a few minutes of exposure versus months and months of true microgravity aboard the International Space Station and low Earth orbit, I don't think there's really a comparison there. So I think in my mind, there's no perfect replicate for what we can do in low Earth orbit.
So short of actually being allowed to go up into orbit, what's kind of in your dream list of things you might want to see or are hoping for in the future? So like if you had an unlimited budget, like what would you really be excited about interrogating or getting into?
Yeah, absolutely. Being able to reproduce some of these initial findings that we've found with our cardiomyocytes in a high throughput way. So basically increasing our N value, as I called it, doing more and more of these experiments more quickly in a more rapid fashion, being able to incorporate robotics into some of this work so that the astronauts wouldn't have to have as much hands-on time, so that perhaps our experiments could be more reproducible, which is, as we're alluding to, an issue that's true across science, not just in space. But of course, robotics is not an easy thing. It's not a cheap thing especially to implement in space, where fluidics and those dynamics are completely different. But yes, if I had a dream, that would be my dream, to ensure that these experiments can be reproduced in a high throughput automated fashion. And I think another element of this is incorporating artificial intelligence. So perhaps figuring out a way so that AI could do some of our experiments for us, so that we would have to do less hands-on experimental manipulation in the day-to-day. Again, if you're giving me an unlimited budget, so I'm running with it, but those are the things that I would incorporate.
So what are the biggest barriers now to progress? Is it money? Is it technical? Is it a combination of both? What's sort of like in your mind, the biggest hurdles?
Yeah, I think it is a combination of things. I think it is the funding initially. I do think that's changing, just because as the commercial area and private space flight is really starting to take off, that problem of funding, I think, is going to get alleviated over time. The federal government, as we've talked about, is funding this area very, very heavily. I think in my mind, it's still technical. It's still being able to reproduce these experiments in a high throughput way, and incorporating some of these elements of robotics and automation and artificial intelligence could help us address that question.
Mm-hmm. And if you could speak to this, I'd love to hear it. What's actually next for your research? Like, what are the things that's on the horizon for you guys?
Yeah, so like I alluded to, we actually have an experiment being launched aboard the Axiom 2 mission in May, and we are hoping to examine the transfection or the introduction of DNA into pluripotent stem cells in microgravity. This is something that hasn't really been done before. We want to see what's the efficiency of this process. How easily can we put DNA into stem cells in a microgravity environment? And is that comparable or even better to what we can do on the ground? And part of the reason we're doing this is to set up our next experiment beyond that, which is hoping to be launched in December, which is actually the full production, the full reprogramming process of induced pluripotent stem cells in microgravity. This is a process that takes about 30 days to do on the ground. But we do know, since there are differences in stem cell proliferation in microgravity, this is worth investigating. Perhaps we can reprogram cells at a pace and an efficiency that's way higher in microgravity than we would be able to on the ground. And that could have implications for the biotech industry as well, since iPS production is still, to this day, an inefficient process, the production of these induced pluripotent stem cells. And if we can get an order of magnitude better in microgravity, then I think that would be very exciting for cell-based therapy and other applications on the ground.
So watching this, how do they get involved in this field? How do they actually get into, a future where they say they work on stem cells in space, or they help design the experiments? Can you kind of walk through somebody what that pathway might look like?
Yeah, I think the good news is because space research and it's because of the commercial spaceflight industry, things are becoming more accessible. It's cheaper to get to space these days. I think the reality is, if you're, say, a junior trainee, a graduate student who wants to get more involved in this work, it's way more easy to do this than it was, say, 20 years ago. There are actually NASA grants that you can apply for that can fund this kind of work. There are even crowdsourced experiments that are being done. Say, for example, folks take imaging experiments in the International Space Station. They're taking pictures of cells and so on. There are certain experiments that can be crowdsourced to the greater audience, to the greater public, so that they can help with some of those experimental analyses. And in fact, I've been involved with certain high school programs who have done some of the data analysis for the experiments that I've launched in the past. So it's much more accessible these days than it is in the past. There are actually specific training programs now, in terms of master's programs, PhD programs, that are incorporating biomedical research in space as part of the training. Certain schools like the University of Colorado have a great program in this. And so I really do think this is an area that's going to become more and more popular, especially when it comes to training in the future.
Wow, that's very exciting. Well, we're at the end of our hour, so I want to thank you, Dr. Arun Sharma, for joining us today. Dr. Arun Sharma is an assistant professor at the Cedars-Sinai Medical Center Regenerative Medicine Institute. I also want to thank our co-sponsors for the series, Harvard Medical School's Center for Bioethics and the Museum of Science. And I want to thank you, our audience, for joining us today. Have a great rest of your day. Thank you for joining.